Hi, welcome for this session, chapter 2, lesson 5, and this is about enzymes. This is about enzymes. When we are looking or talking about enzymes, something must be clicking into our minds. And how do these really work? Maybe. So enzymes are, first of all, they are proteins. They are made of protein. Made of proteins. So they are proteins. Because they are proteins, they will have some drawbacks which we will be looking at later on. But of course, in living systems, these are the best that we have. In living system, these are the best that we have. So enzymes are catalysts for chemical reactions in living things. They are catalysts. They are catalysts for chemical reactions in living things. When we say they are catalysts, we mean they speed up things. They speed up reactions. So we will be looking at how they do that. We will be looking at how they do that. The main ideas here are catalyst lower activation energy. That is one way by which it will speed up chemical reaction by lowering, of course, by lowering the activation energy. And the second one is enzymes allow chemical reactions to occur under tightly controlled conditions. So because of this also speeds up the reaction. Think about life, for example. Think about a Venus flytrap. Think about a Venus flytrap. A Venus flytrap. Think about a Venus flytrap digesting a frog. How does it do this? This is a plant and it's digesting an entire frog. So it happens through the activation of proteins. So the action of proteins called enzymes. So it is through the action of proteins, and these proteins are, of course, enzymes. So enzymes are very important. So enzymes helps to start and run chemical reactions in living things. Not only to run, but even to start it. Okay? So for example, enzymes are needed to break down food in small molecules that cells cannot use. That cells cannot use. So without enzyme, for example, the Venus flytrap couldn't break down its food either. That is, it couldn't break its food either. That is the frog. So it couldn't break the frog if there was no, of course, enzymes which will carry out these reactions. So a catalyst lowers activation energy. That is what we will base ourselves as in our discussion today. We will be looking at a catalyst. A catalyst lowers activation energy. A catalyst lowers activation. So remember that you learned about activation energy in the previous section, but as being maybe pushing a rock up a hill. That is what we were looking at. Like pushing a rock up a hill, that was what giving it a push. So when enough energy is added to get that rock to the hill, then the rock can roll down by itself. So activation energy gives similar push to chemical reactions. So once reactions start, it continues by itself. So what activation energy does, activation energy gives reactions a push or a start. So without activation energy, then things will not work as we expect. And normally, chemical reactions will continue by themselves. So often the activation energy for chemical reactions comes from an increase in temperature. Often the activation energy for a chemical reaction for a chemical reaction starts for a chemical reactions starts or comes from an increase comes from an increase comes from an increase in temperature. It comes from an increase in temperature. So the reactants may not interact enough, or they may not be high enough, so at high enough concentration to quickly form the products of the reactants. I mean here the reactants may not interact enough, or they may not be at a high concentration. So we take an example. If we are having particles here. They are at low concentration. So for them to interact, we increase temperature. So temperature gives them a push. This is just but an example. So temperature will give them a push to quickly form products. However, both the activation energy and the rate of chemical reaction can be changed by a catalyst. So the both can be changed by a catalyst. So what we are saying here, a chemical reaction can be changed by a catalyst. So it can be changed by a catalyst. In our case, what are we having here as a catalyst? So what we are having here as a catalyst are enzymes. So we are saying here that what they do, they decrease they decrease the activation energy needed to start a chemical reaction. So catalysts decrease the activation energy. They decrease the activation energy needed to start a chemical reaction. Needed to start a chemical reaction, and as a result increases the rate of chemical reaction, of course. and as a result, sorry, and as a result, increases the rate of chemical reaction. So by lowering the activation energy, they increase the rate of chemical reaction. If we look at what we have here, let me try to make it bigger a little bit so that it's clear. So catalysts and activation energy. Catalysts and activation energy. Catalysts and activation energy. So under normal condition, a certain amount of activation energy, under normal condition, a certain amount of activation energy is needed to start a chemical reaction. So a catalyst decreases the activation energy needed. That is what a catalyst does. It decreases the activation energy needed. If we look at our diagram here, as we move, these are the reactants. This is we are raising our ball to achieve the activation. This is the activation energy here. This is the normal activation energy. But what happens is that uh, in the presence of a catalyst, that is a catalyzed reaction, then this is the activation energy. It means the graph shifts from where it was up there and comes down here. So the graph shifts from up here and comes down here. So you can see this difference is what we are calling reduction of the activation energy. So the activation energy is decreased. So describe two functions of catalysts in chemical reaction. One, they reduce. They reduce or they decrease the activation energy, that is. They decrease the activation energy. They decrease the activation energy. And apart from that, they also are responsible for speeding up the reaction. They also speed the reaction. So these are the two main things that they will do. One, they will speed the reaction, and they do this by lowering the activation energy. So under normal condition, the reaction requires a certain amount of activation energy, and it occurs at a certain rate. So it can never occur unless there is an activation energy. So when a catalyst is present, it lessens the energy needed and the products to form faster. So although catalysts take part in chemical reaction, Catalysts are not considered to be either reactants or products because catalysts are not changed or used during reactions. So catalysts are not changed or used during chemical reactions. So we are going to look at how, of course, the catalyst that we have here works, or the catalyst that we are talking about. So the catalyst we are talking about here is nothing but enzyme. So here, uh, this is how an enzyme works. So enzymes, they are catalysts. So if you look at them here, you will see that we are having here the substrate. So substrate is what is going to be catalyzed, or what is going to undergo chemical reaction. So the reactants. So the reactants are the substrate. Then we have enzymes. This blue stands for enzyme, and these two here, they stand for reactants, and of course, this here stands for product. So this is product. These are the reactants. So the reactants will give us the products. But how does this happen? So substrates bind to an enzyme at a certain place called the active site. They bind to them at a certain place called the binding site. So at the binding site. Step number two, this is what happens. The enzyme brings substrate together and breaks their bond. Very important. It has to bring them together, and after bringing them together, it breaks their bond, and in the last or final stage, the catalyzed reaction forms products that is released from the enzyme. So it will be released from the enzyme, and the enzyme will be there for another reaction. So let's look at how these, we'll be looking at just an example of how this can occur. It is kiddish, but it's easy to understand. So if we take this to be the enzyme, and these are the substrates, so you have to bring these together. Then this is the active site. If enzymes are specific, enzymes are specific. They have specific sites. You cannot put this ball there. But these ones, they fit exactly because these enzymes, as we say here, enzymes are specific. By specific, I mean they carry out specific reactions and they don't do everything. Like an enzyme that digests carbohydrates in the mouth, carbohydrates in the mouth, that is amylase, will not do any other but only breaking down carbohydrates. It will not be responsible for making proteins. It will not be responsible for breaking down sugar and so forth. So it is specific. That is what we are trying to show here, that this ball may not fit there. So they will bring them together, as you can see. Then this now will form a bond. This is now the bond. After the bond has been formed, of course, this will act like a bond. After the bond has been formed, what happens is that we are having our product here and the enzyme is there for another reaction. So chemical reactions in organisms have to take place in an organism's body. They have to take place in an organism's body because the reactions must take place very quickly. They usually need a catalyst. They usually need a catalyst. So enzymes are catalysts for chemical reactions in living things. So enzymes, like other catalysts, lower the activation energy. Enzymes, like other catalysts, lower the activation energy. Like other catalysts, lower the activation energy. So they will lower the activation energy. In reactions that are reversible, such as carbon dioxide and carbonic acid that we saw previously, enzymes do not affect chemical equilibrium. So enzymes do not affect chemical equilibrium. They do not affect chemical equilibrium. What I mean here, uh, enzymes are, this means that the enzymes do not change the direction of a reaction. They just change the amount of time. Okay? They don't change, they don't change the direction of a reaction. So enzymes do not change the direction of a reaction. They just change the amount of time needed. They just change the amount of time needed. So the amount of time needed, of course, for the equilibrium to be reached. For the equilibrium to be reached. For the equilibrium to be reached. So enzymes are involved in almost every processes in organisms. From breaking down of food to building proteins, enzymes are needed. For example, as we saw earlier, amylase is responsible for breaking down starch, and we have other millions of enzymes which are responsible. So if we take an example of amylase, if we are looking at uh, how if we are looking at how, for example, if we are looking at how, so almost all enzymes are proteins. Are proteins. So like other proteins, enzymes also have problems By problems, I mean that they have their own drawbacks. So each enzyme depends on the structure of amino acids. So they depend on the structure of amino acids. So as we saw earlier, proteins work like uh, they work like this model of the lock and key. The lock and key model. The lock and key model. So when we are looking at the lock and key model here, what happens is that uh, things, enzymes, have to fit exactly to these active sites, like a key and like a key and like a key and lock. So like key and lock. So after that, of course, bonds will be formed and the two will be joined together and then they will be released from the site for other structures to continue with their reaction. So we are saying here that anything that will affect the shape, so anything that will affect the shape of an enzyme will affect the rate of reaction. It will affect the rate of reaction in that if, for example, some things like temperature, temperature, pH, and other inhibitors will affect enzyme action. If we take an example of temperature, as temperature increases, it denatures proteins. So proteins will be denatured. If proteins are denatured, they lose shape. They lose, sorry, they will lose their shape, and as they lose their shape, they will not be able to carry out their function because any alteration in the shape will directly affect the function of the protein or the function of the enzyme in this case. So we are looking at these things as being very serious in terms of affecting the rate of reaction. So anything that alters or affects the shape of an enzyme will also affect the rate of reaction, and this happens directly. So we will stop here for today, and before we stop, we can look at, we can stop here. We will be looking at something else in the next video, inshallah.